Okay, so hello again. Welcome to the Unit 3 review covering the immune system. Um, Today I'm just covering some of the main things with the immune system, mostly first line, second line, third line defense, as long as, or sorry, as also trying to kind of show you how you can work through your case studies. If I lose my voice a little bit, I apologize. It's very phlegmy today. I'm not sure if I'm sick or not. Probably I am. It's that time of the year, but I'll try to get through it. If you have any questions, feel free to just throw it in the chat. You can also message me after this if you're watching the recording. So the overview, like I mentioned for Unit 3 review, we're going over the first, second, and third line defense, and then we'll be talking about how to work through the case studies. So first line defense. So first line defense is our first line of defense to prevent infection or illness. As you can see, I have it broken down between the physical and the chemical barriers. Some of the physical barriers include our epidermidis and the dermis, our mucous membranes, our mucus, cilia, coughing, sneezing, blinking if you're in the eyes, and then vomiting, diarrhea for the GI tract. You can kind of see that most of your physical barriers are roughly the same, involving mucous membranes, mucus, and cilia in various parts of the body. But then you have very specific things like eyes blinking or diarrhea to vomiting for the GI tract. So if you're given a case study or a question on the exam regarding the first line defense, make sure that you read through which organ or organ system is being discussed. And then from there, you can determine what first line physical barriers there are. Again, for the chemical barriers, you can see how our skin has acidic pH. It's salty and it's also oily. We do have lysozymes and lactoferrins in the respiratory system. The eyes are unique that they also have lacrimation or their tears. The tears actually have some type of antibiotic protection. And then the GI tract, also mucus, saliva, hydrochloric acid, bile, different enzymes, and gastroferrins. And the reproductive system, mucus, acidic pH, and lactoferrins. Again, same thing. If you get asked about chemical barriers, see which organ system we're talking about. Most likely the question is going to ask you about physical and chemical. So maybe something on the lines of, give me two physical and two chemical barriers for the reproductive system. And then you just usually type those out, or it might be one of those multiple choice select all questions. Resident microbes, again, remember why we have resident microbes and what kind of resident microbes we have. I believe Dr. Cotter asked you all to at least have one microbe per organ or organ system. I recommend just reviewing some of these, like if you get asked for resident microbe, the nose, Staph aureus is a pretty common one. The skin, Staph epidermidis. I think we mentioned Streptococcus pneumoniae for the throat. In the intestines or the GI tract, we've got Escherichia coli. In the vaginal tract, if you're a female, you might also have Candida albicans. Um, and then another thing to note for a question like this would be the reasons why we have our normal flora and what they do for us. So a question would be asking, give a resident microbe for this system and give us some reasons why this resident microbe is helpful for us. Giselle, you said, where would sebum be on the first line of defense? Good question. I'll just go back one. So sebum is considered a chemical barrier. That is the bactericidal acids or our oils. Sebum is included in that. So it would be a chemical barrier for us. There are some physical properties to oil, kind of how it traps, it can use it traps microbes and whatnot, but the primary thing is the oil and its chemical properties are what protect our skin and reduce the activity of an infection. Good question. Okay. Second line defense. So here's where people or students tend to start struggling. Um, Recall that in every immune response, macrophages and neutrophils will be involved. The questions that are going to be asked on the exam will probably ask you, what two um, immune cells are always involved, and then they might ask you for a third immune cell that's involved based on this specific um, infection. And so neutrophils, macrophages should always be in your answers if it's asking you for any second line immune cell. And then remember that basophils or mast cells are used for allergic responses, depending on where the allergic response is occurring. And then eosinophils primarily are used for parasitic infections. Um, Recall that in the chapter 15 and 16 case study, you were asked to write down the cells, the immune chemicals, and the immune processes. I'm just going to tell you, you're probably going to have similar questions on the exam as well, asking you if you write macrophage, give me some of the immune chemicals and the immune processes that's involved in. You can also flip this and kind of ask a question along the lines of what second line chemicals are primarily uh, part of the phagocytosis in the body, or what actually forms pus. Neutrophils form pus. So things like that, being able to take the immune chemicals, the processes, and the cells, know which ones align, and that way, no matter how the question gets asked, you know how to respond it properly. Third line, and this is the fun one, the one that we spent, I think, the last 2 days working on, but still the one that gets students the most. So recall that we have two different types of third line responses, depending on whether if the infection is either inside of our cells or outside of our cells, like in our blood vessels or in the skin. So if you are inside the cell, or an intracellular response, or a cell-mediated response, you are looking at the Th1 CD4 helper T cell. So I wrote down very specific steps because the way the questions might ask you, based on what I've seen before, is okay, which helper T cells involved, Th1 or Th2? Which cells are activated? Cytotoxic T cells, memory T cells, or plasma B and memory B cells? And then which receptors are actually involved in the infection? So if we're talking about any of the T cells, the CD4 helper T cell, the cytotoxic T cell, the memory T cells, they use T cell receptors or TCR. I'm going to harp on this a little bit because we don't talk about the T cell receptor a lot in class, but I do recall this question always gets asked on the exams. It might not be on this one, but over the last 5 years, I've seen at least one question asking about receptors, and I'm just giving you the abbreviation. T cell receptors, or TCR, are used by the T cells. So repeat that to yourself. It shouldn't be too difficult to recall, but I sometimes see students throw other receptors in here, like the Th1, they'll throw in CD4, they'll throw in a bunch of other receptors that we don't really talk about, MHC1 and 2. Just remember that the T cells use T cell receptors to screen the other cells. And then the plasma B cells or the memory B cells use B cell receptors or BCRs. So again, back to the first scenario, if you are an intracellular response or a cell-mediated response, or it's inside your cells, you are the number one, so you're going to use Th1 helper T cells using the CD4 plus receptor. This will then activate the CD8 cytotoxic T cells and the memory T cells. These T cells will then use the T cell receptors to go and start screening, checking all of the cells. Remember that MHC1, making sure that the cells are okay and they're healthy and that they're your cells, or to see if there's any abnormalities, either genetic mutations in the MHC1, or sometimes if it's a virus, sometimes the viral spike protrudes, and that's what gets screened by the T cell. If the T cell finds a virally infected cell, it's going to use perforins and granzymes to destroy it. It's going to use perforins to create small holes or ruptures in the membrane, and then it'll use the granzymes to actually start digesting all the pieces of the virus and are fortunately our destroyed cell as well. Now, if the infection is outside of our cell, sorry, so the first one for the inside the cell, predominantly we're talking about viruses. Um, Anything other than a virus, a bacteria, a parasite, a fungal infection, an allergy, that's all outside of your cells. So that's going to be an outside the cell extracellular response, or what could also be called a humoral response. Remember, your humors are your liquids. In this case, when we talk about humoral response, we're talking about the blood. So anything that's outside the cell is going to be part of a humoral response or extracellular response. And again, I'm saying all the different words because they could be said different ways on the exam. I just want to make sure that you make the connection between humoral, outside, extracellular, and cellular, intracellular, or inside the cell. Those are all the different categories. So if it's outside the cell, the macrophages will then report to the Th2 helper T cell using the CD4 plus receptor. The helper T cell will then activate plasma B cells and memory B cells. The B cells will use BCRs or B cell receptors to start screening for the invading microbe, the bacteria, parasite, etc. And then the difference between the T cells and the B cells is that the B cells will actually secrete antibodies to start following through these four processes of agglutination, neutralization, optimization, and complement fixation. The key difference between the two is that antibodies do not destroy or kill any invading microbe. They're only there to attach to it, disable its ability to attach to other things like our tissues, season the microbe by optimization, making it more flavorful for the macrophages to come eat. And then complement fixation. I'm not going to get into complement fixation too much because we don't talk about it much, but it's a bunch of other little things called complements that help to destroy that cell. Um, and then recall that we have different types of antibodies that are secreted through the body. So I would definitely know which antibody, the different antibody types and where those antibodies are involved. So remember the IgM is our massive cell, our biggest antibody. That one's always released, but it doesn't leave anywhere outside the blood. It stays primarily in the blood. Our IgG or our go anywhere antibody that can travel across the blood barrier into the tissues. It can also cross into the placenta. Our IgA is primarily involved in mucosal infections such as the respiratory system, the GI system, and then occasionally reproductive as well. And then IgE is primarily released involved in allergic responses. And those are the ones that we don't like because it gives us that nice little itch allergic reaction. Again, you're going to be asked, I know at least a question or two about the antibodies. More so, what it might ask you is, in this case, they gave you let's say a respiratory system infection. What kind of antibodies would have been released? And then you would say, oh, IgM and IgA and IgG, for example. Does anyone have questions about this one? Okay, not. So this exam is going to break down very similar to our case studies. Again, the questions may not be type in, they could be multiple choice, select all that apply, or short answer. But the way you want to tackle a case study is first question, ask yourself, where's the infection and what could be causing it? So where in the infection? We're talking, again, respiratory, reproductive, GI tract, skin. I don't believe she's going to give you a skin question. That's typically a little easy. I believe she's primarily going to focus on reproductive, GI tract, and respiratory system. And then what's causing it? Is it bacterial, viral, parasitic, etc.? So once you've determined that, in this case, I use the example, okay, I have a bacterial infection in the vagina. This is usually Candida albicans. but I'll just throw that one out for this one. Um, List your first line chemicals and the first line chemical and physical barriers. So then your next step, all right, what physical barriers do I have in the vagina? I got mucous membranes, I got mucus, I got cilia. What about chemical barriers? My mucus, acidic pH, and lactoferrins. Okay, first line taken care of. Step three, do I have any resident microbes in the vagina? Like I mentioned before, we've got Candida albicans. You also got Lactobacillus acidophilus. Acidophilus. Again, you don't have to know multiple. I would just know one, maybe two if you want to have a second backup. But just list all your residents. Step four, start listing through your second line cells that could be involved in this. In this case, I wrote macrophages and neutrophils. List the chemicals, digestive enzymes, interleukin 1, interleukin 6, and complement proteins C3b, C3a, and 5a, C5 through C9. And then what processes are involved, phagocytosis, fever, inflammation, and the membrane attack complex. And then step five, this goes on your third line, is the infection inside or outside your cell. In this case, I said the vaginal infection is a bacteria outside the cell, meaning it's an extracellular response or a humoral response, which will require a Th2 helper T cell, B cells, and antibodies. You can take any infection, any location, and the bacteria or the microbe that's causing it, and you can break it down into what barriers, what first, second, third line barriers, what residents are involved. You'll do very well on this exam. Just other things to bring up for this exam. Again, remember your signs of inflammation. Your big four, your tumor, calor, dolor, uh, tumor, calor, dolor. I'm forgetting the fourth one, but I'll let you guys look that one up. Rubor. Um, Being able to know what those are, the definitions for those. Let's see what else is in here. Fever. Why is fever important? What does it help us do in the body? How does it promote our immune system and also weaken uh, an infection or an external infection? Um, you can ask a question about the like different types of immunity, like natural passive, natural active, artificial passive, but you might only get like one or two questions on that. I wouldn't focus too much on that one if I were you, but it's pretty straightforward. Um, the types of hypersensitivities, I would look at that, make sure you understand the difference between an immediate, cytotoxic, an immune complex, and delayed hypersensitivity. Again, if we haven't covered it, you might have covered it today. If you're in the Monday group or Wednesday, we'll probably cover that in Wednesday's group. That's basically it. What I would recommend is for your case or your study guide, obviously most of it should be filled out, but if you haven't done so, I would at least have pages 8 and pages 9 filled out because those would have majority of the information regarding the first, second, third line responses. And then you also have some practice cases on page 9 that show like very similar to how I broke it down here, how you have, okay, list the first line physical, first line chemical, resident flora, second line, et cetera, et cetera, et cetera. But if you know nothing else besides, I know how to go through my first line, second line, third line, depending on the infection and where it's located. And if you didn't study anything else, you'd probably still get an 80% on this exam. It's going to be very heavy on these types of questions. And you can see how in this case study, I could probably make, 1, 2, 3, 4, 5, 6, 7, 8. I could probably make 8 or 9 questions just in this case study alone. Um, so if you study that, I think you'll all do well. If you have any questions about what we went over, actually, does anyone have any questions about anything that we went over for the people in the chat? If not, like always, feel free to reach out and message me with questions. The exam will open just like normal. Really, it's Thursday morning, and you'll have until Saturday evening to complete it. I'll send out those details in an announcement as well. And then if you need feedback on the practice cases on page 9, just go and work through them, send them to me, and I'll send you the answers essentially for that one. That way you at least have that. This goes through 3 different types of infections: a GI tract with a parasite, bacterial infection across the placenta, and a respiratory infection with the influenza virus. So those are very good practice cases for you to practice before we get to the exam. If no one else has any questions, then I'm going to go ahead and wrap this one up. Good luck on your exam. Reach out if you have any questions. But other than that, that's basically it.
Uh, Luis, I actually did have a question.
Yeah, what's up, Giselle?
On page 8 of the study guide,
Hmm.
can you kind of go over the suppressor T cells?
Yeah. Okay, so we didn't talk about the suppressor T cells a lot. So,
Yeah.
um, and she probably won't ask it, but basically what suppressor T cells are is you've got the macrophage taking the piece of the antigen of the bacteria, virus, whatever. It goes to the CD4 plus general. It at that process starts the third line defense. And so the CD4 generals will then activate the B cells or T cells. In this case, we'll just go with T cells. You have your cytotoxic T cell, the memory T cells, they'll start killing everything. What the suppressor T cells are is that there's a feedback, kind of like a feedback mechanism back to your body saying that, hey, I think we've got this infection under control. You can probably stop calling on more T cells to come out. So the suppressor T cells will then be activated to basically stop all the other active T cells from doing their job. Basically telling them, all right, guys, we're done. Wrap it up. Stop killing what you're killing. We're good to go. Just stop doing what you're doing and then go back and get recycled somewhere else in the body in the I forgot where the recycling center is at. But essentially it's the T cell that stops the third line response to the body. So where you could have issues is if your suppressor T cells aren't working, your cytotoxic T cells will keep searching for an infection when there isn't one, which you think would be a good idea. But if you think about how your body reacts when it's ill, it starts taking away resources from other parts of your body to promote and make more cytotoxic T cells and immune cells to keep fighting an infection that doesn't exist. It could actually make you very sick because your body's no longer acting as it should. Your body's always thinking it's infected, under attack, and you're not going to get the right resources. You're never going to be energetic, have your energy back. You're just going to feel tired, lethargic the entire time. That's a little bit extra that's needed, but essentially suppressor T cells, they're third line specific cells that ultimately they'll stop the immune response would be the quick the quick and easy way to say it.
Okay, thank you so much.
Yeah, no problem. Good question. Okay. Any other questions? Okay. Again, good luck with your exam. Reach out if you have any questions. But other than that, enjoy the rest of your night and good luck.
Thank you, Luis. Have a good night.
Thanks, you too.